Hello, everyone. Today, I'm going to talk about our automated platform to monitor in real time and dye-free the expansion of your cells in a bioreactor. This is a joint presentation with Bristol Myers Squibb. My name is Émilie Vier. I'm the former Chief Executive Officer at Ovizio Imaging System, and I'm sharing today the floor with Ivy Erufa from BMS. She will present how our iLine platform has been integrated into the CAR-T process and which value it has brought. Ovizio Imaging System is a Belgian company with 10 years experience in developing label-free platform from the software to the hardware part. We have established mainly our footprint in the bioprocessing market, especially in the cell and gene therapy and the recombinant protein production. Why Ovizio? So perhaps I should start at the beginning of this story. So it came from an observation, that all the labs growing cells face bottleneck challenges when scaling up their activities, from the sampling to the preparation, staining, reading, and analyzing. It requires human action, which may, of course, induce a risk of contamination, variability, or bias in the cells measurements, such as cell counting and assessing viability. That's why we decided to develop the iLine microscope, which is an automated platform, dye-free, that brings automation in your cell's expansion process. The cells will traffic from the bioreactor to the microscope in the closed loop. All over your cultures, you get access to real-time monitoring of the cell's attributes, cell counting, viability, and much more. This is an automated solution, completely dye-free. As you can see on the left side, the microscope is connecting to a rocking motion platform. And I would mention that on top of that, this summer, we will launch the first cGMP version of the microscope to comply with any first-in-human clinical trials or manufacturing operation. Our CONNEXION is bioreactor agnostic. In fact, we can connect with 90% of the bioreactors in the market, such as small scale, single use, rocking motion, benchtop, or stainless steel bioreactors through different types: welding, lower locks, or Novacemtum sterile connection. The BioConnect is a single use and very versatile disposable. What finally will you get from the iLine microscope? So such an images like that, as you can clearly see now on the slide. We have built, as of today, several algorithms to get access to our customers, how performant are their cultures. We can assess viability, do cell counting, such as CAR-T cells, TCR T cells, NK cells. We can track any objects in the cell culture, such as beads, red blood cells, different subtypes, of course. And then we can either classify and segment properly the change of a state in a cell, such as activation status or exhaustion. Now, to give my words more concrete, I will leave the floor to Ivy Erufa from BMS.
For that introduction as well. So for the rest of the time, I will be talking about the automation of T-cell expansion using the iLine platform. So as many of you are well aware of, process analytical technologies, or PAT, consist of designing, analyzing and controlling manufacturing through timely measurements for critical quality attributes or performance attributes of raw and in-process material and processes, really with the goal of really ensuring final product quality. So PATs should really provide real-time assessments, in cell therapy for various critical process parameters, such as viable cell concentration, media metabolites, or nutrients, pH and DO are various examples. And it's also key to include that new PAT for cell therapy should really work to provide understanding for critical quality attributes, such as cell phenotype, cell functionality, as well as cell health. Additionally, we anticipate that cell therapy PET platforms should really provide real-time monitoring of these attributes. They should be able to interface with bioreactors or any other cell therapy processing equipment. They should also interface directly with a control automation system, as well as utilize pre-sterilized single-use contact surfaces. So these should be disposable type systems. Additionally, we anticipate that these platforms should be, non-invasive type sampling and also non-destructive analysis, since it's well understood that the material that we are processing are our patient cells, that the cellular material is limited, and so non-invasive and non-destructive sampling is preferred. So within the cell therapy process, there's the expansion step within that overall manufacturing process, and it is well understood that this process is quite manual. So we have here on the right side of the slide just a breakdown of examples of operations that manufacturing staff would have to undergo just to be able to attain a sample from their bioreactor, and therefore make modifications to the bioreactor recipe to be able to change the feed rates that the bioreactor may be undergoing. And so this requires operators to go into the lab or to the GMP suite to gown up and perform these operations stated here. Currently, majority of our operations are quite manual, as you can see from the right side of the screen, with a limited number of automated steps. And so to improve this workflow, we worked and we identified the iLine platform shown here. The iLine, as Emily described, is an inline cell counting system. It's also a dye-free approach for counting cells. Essentially, how the system works is that the instrument pulls material using a membrane pump from the bioreactor. The cells are then circulated through the white sampling loop shown on the screen and inserted into the iLine device. The cells are then imaged and returned back into the bioreactor. So this provides a closed cell processing platform. So this enables the removal of the risk of bioreactor contamination due to sampling steps. Additionally, the system is also bioreactor agnostic, and so it can interface both with rocking motion systems or various stir tank platforms. Additionally, the system utilizes OPC UA and web services capabilities, and so the system is capable of interfacing with process control systems that various manufacturing sites may have to enable automated CAR-T bioreactor operation. So generally how the system works is that there's a image acquisition phase followed by an image processing and image analysis phase. So for the most part, the instrument performs several captures of cells as they are flowing through the single-use BioConnect system. And it captures not just a two-dimensional image of the cells like traditional microscopes, but captures a three-dimensional hologram of the cells with the imaging height of about 120 microns. The operating software called OS1 then displays the cells within your image field of view, which is extremely convenient as a experimentalist, that you're able to actually visualize the state of your cells while having real-time display of viability as well as viable cell density on the right side of your screen. After image acquisition is then followed by an image analysis phase. It's very interesting that this is all performed on a single cell level. So the software OS1 takes the acquired images and segments each cells and analyzes each of the segmented images. And as Emily discussed previously, the software then classifies cells as either viable, which I have as green, the green arrow pointing to, either dead, which is the red arrow. The system is also able to capture debris as well as cellular clusters within within your image. Additionally, the software itself then also overlays these classifications, as you can see on the screen, and so you can have, again, a great way of visualizing the state of your culture. So we, as a company, have executed over 250 bioreactor expansion runs leveraging the LNF platform. I'm showing examples of just 7 general experiments that we performed. As you can see with the data set on the left here, we have very good correlation with the LNF with our reference cell counter. Additionally, with the graph on the right, one great benefit of the LNF is the amount of resolution that you attain into your culture. Typically, you may sample bioreactors maybe at a frequency of once to twice a day. However, this system provides you with hourly assessments of your culture, and so you have a lot more process understanding with this platform. Additionally, within our internal development efforts, we've demonstrated that the LNF has or displays good performance with multiple cell therapy processes, and so we've determined that so far the system is process agnostic, and so with various T-cell products that various companies would be working with, we've noticed that there isn't the same configuration applies to multiple processes. Additionally, as you see with the graph on the left, we do see very good comparability with our reference cell counter, and we've also seen no negative impact to our cell therapy processes, and so the inclusion of the LNF does not impact our VCC, our viability, or cellular phenotype or functionality so far in any way. Additionally, we have successfully been able to execute automated experiments with the LNF. So the main benefit of the LNF is not only to provide inline measurements of viable cell concentration, but it's to therefore allow this cell counter to interface with a bioreactor system to be able to therefore drive the full CAR-T expansion process. So we have internally executed successfully over 75 fully automated bioreactor expansion runs. The graph on the left is an example of a process that one of our earlier processes that we performed, where we performed daily sampling and changed our bioreactor feed rates, which are indicated with the arrows here at the different points that we changed our feed rates. And as you can see, the automated system, which is here denoted in pink, cells grew substantially faster than cells that were executed with manual operation. The main difference is that when the bioreactor reached the target viable cell concentration to initiate the next bioreactor feed change, we're able to actually do that transition in an automated fashion rather than waiting to the next sampling operator sampling step. And so what we learned from this initial experiment was that automated expansion may improve growth kinetics and really potentially reduce some process variability that is experienced through timely media feed adjustments. Additionally, we did also perform analytics to demonstrate impact to cell functionality and cell health. And we saw that there were essentially zero impact to, although the cells grew faster, differentiation state or overall cell phenotype remained unchanged. So we really just saw an improvement in growth trajectories of our cells. So compared to the earlier slide that I showed with very more manual steps where we have operators having to execute a series of steps to enable feed changes with our bioreactors. What the iLine offers is the ability for operators to really just inoculate our bioreactors at the beginning of our expansion process, and then walk away and until harvest is required. We are able to view throughout our entire expansion process the state of our expansion. And so although we're not necessarily having to come into the lab or come into our GMP suites, we still have full visibility with the imaging segment, as well as the viable cell concentration and viability segments of the system. And so we're able to execute, remove all of our manual steps from our expansion unit operation and streamline these with fully automated steps. And just finally, I do want to highlight the benefits of the iLine for cell therapy expansion. And these are really to improve the process and product understanding through real-time monitoring. This is key for cell therapy, as we are still in a very much a learning stage of this process. And so being able to have high resolution data to really help us build understanding of the trajectories that our cells are undergoing within our bioreactors. Additionally, the system does reduce process and product variability through utilizing feedback process control. Additionally, the system does promote closed cell therapy processing. As you can see, there are no open steps. There's no open sampling. All steps are performed in a closed environment. The iLine also promotes the reduction of cost of goods within both development and manufacturing. If you're not having operators having to fully gown up to enter GMP suites to execute sampling and bioreactor feed changes, this provides a significant improvement in the costs associated with cell therapy expansion. And finally, the iLine enables the acceleration of process development and characterization. Again, if we have higher resolution understanding to growth trajectories, we're able to accelerate our development efforts as well as our process characterization.